Welcome back. Let us look at an interesting application of graph theoretic approaches again. Right. So, we have looked at a bunch of applications so far, but today we look at a very interesting and emerging field, that of microbiome systems biology, and how we can, building on concepts we learned in the previous class on representing metabolic networks and so on, how can we extend such approaches to modeling microbial interactions in communities, right, or we call them as communities or consortia or microbiomes. Typically microbiomes are very large, communities could be smaller but generally used interchangeably. So, how do we study microbial interactions in these kinds of scenarios? By using graph theoretic approaches. So, let us look at what are microbial communities. Microorganisms are ubiquitous, right, but they rarely exist in isolation. They often thrive in a multi-species dynamic environment, and the community structure itself is driven by a complex web of interactions. You have metabolic interactions that are predominating, right. There are all kinds of interactions that exist, but metabolic interactions have been identified to predominate in many of these cases. And there are many microbiomes that have become extremely interesting for various reasons. These include the soil, gut, which we will try to focus on a little bit more today, or the skin, clean room microbiomes, and built up environment microbiomes, and so on and so forth. Microbial consortia or communities have also been useful in metabolic engineering applications, and understanding these interactions between microbes is crucial to both understand and manipulate environments and microbiomes such as the gut, and also to develop microbial consortia for biotechnological applications and so on. So, these are all very interesting as we embark on trying to figure out how to understand the interactions, the metabolic interactions between microbes in various milieu like the gut microbiome. The gut microbiome itself is a very complex ecosystem. For example, this was a very famous cover page of Nature which says it is our other genome, right, because the gut bacteria in the body, the gut microbes in the body outnumber human cells by several factors, right. So, anywhere between 3 and 5 and sometimes even as many as 10 has been estimated, but I think the current accepted number is about 3 to 5 times the number of human cells is the number of microbial cells in the body. And the composition of this gut microbiome is highly variable. It depends upon the stage of life, diet, environmental exposure, antibiotic usage and so on. And antibiotic usage is basically a hidden pandemic. Antibiotic resistance is a hidden pandemic that has emerged because of, for various reasons, A, because there is a lot of abuse of antibiotics, and B, because microbes are really smart, they evolve very fast and acquire resistance as soon as they are exposed to a new antibiotic. But these are major challenges that we need to counter, and to counter these we need to understand a little bit more about what are the microbes in the gut and how do they interact with each other and so on. So, the question for us today is how can we understand microbial interactions in communities such as in the gut? Can we use metabolic models similar to what we discussed in the previous class? And how do we adapt these metabolic models to work on large microbial communities and community metabolic networks and so on? And you are familiar with this kind of a picture. There are these large metabolic networks that have been reconstructed for various organisms. We call them genome scale metabolic networks because they try to capture all the reactions happening in the entire organism, right, at the scale of the genome. All the enzymes known are typically accounted for as far as possible. Essentially a comprehensive picture of what kind of metabolic reactions are known to occur within the cell, and there are these draft reconstructions where the stoichiometry or other things may not be very carefully looked at, but they can be readily obtained from databases such as Model Seed, Biopsych. There is also this database called Virtual Metabolic Human today and so on, and all of these databases are very valuable in terms of giving us draft models that can be readily used for metabolic modeling. And there are many methods that exist to analyze these networks. These include constraint based methods and also graph theoretic methods and so on. We will focus today on graph theoretic methods. But what can genome scale metabolic models tell us? This is a very classic paper from several years ago where they talk about how about 250 studies had been carried out with the E. coli model alone and what kind of questions they were used to answer. So, this includes only a small fraction of interspecies interaction studies, which is the major focus for today's discussion. But if you see, typically there are a lot of studies that focus on analysis of biological network properties or metabolic engineering, followed by prediction of cellular phenotypes and so on. So, these are some of the most important applications, and of course model driven drug discovery, model driven discovery and so forth. But what is most important for us is how you can model interactions between species using microbial metabolic networks and models of these microbial metabolic networks. How do we analyze genome scale metabolic networks? There are two broad families of methods. You can also use ODE based modeling, but that is far more challenging, especially to do it at a system scale, because there are lots of parameters that need to be estimated and so on. So, we almost do not consider ODE based modeling when you talk about modeling very large systems, although there are attempts that have been made, successful ones too. Constraint based modeling is very popular for applications such as metabolic engineering, because it tries to understand the growth of a cell at steady state, and this is very useful in trying to predict metabolic engineering strategies and so on. But this demands well curated models. On the other hand, graph theoretic approaches typically focus on path finding. Right? You have a complex graph, and how do you find paths from one node to another, right? This could be one metabolite, and how do you find a path to a target metabolite of interest, which is important for the cell's survival or whatever. And can you also predict new pathways based on reaction rules or atom-atom mapping and so on. So, this is retrosynthesis. This is an example that we discussed a few classes ago as well. And we need methods that are scalable and accurate and can figure all possible routes that exist within the cell. So, this is a sort of wish list for us. So, how do we build scalable methods and accurate methods for studying really large and complex metabolic networks? We need them to be scalable because a single organism's metabolic network is large enough, but then you are talking about putting a lot of these kinds of networks together. There are several organisms in the body, and we typically still build models for only about a handful of them together. Models are available for hundreds of organisms or even more than 1,000 organisms, but we only try to put like 20 or 50 of them together at a time and study, because it gets very intractable otherwise typically when you start putting such large metabolic networks together and try to model collectively what is it that they are up to. So, how do we model microbial communities? There are again many methods to model microbial communities. There are constraint based methods that are applied to single metabolic networks that are extended very nicely to microbial communities. There are graph based methods which we can extend to microbial communities. That is what I will focus on, as the central focus of this course is biological networks. And there are also population based and agent based methods and so on. And there are still a few challenges in terms of the quality of the reconstructions and the compatibility of different models that are curated by different groups and so on, which is because of the lack of adherence to standards and things like that. But I will not get into all of those things, because we will focus mainly on models that are available from excellent databases such as Agora or Virtual Metabolic Human, which are pretty much compliant and easy to merge and very compatible with one another if you want to look at metabolic exchanges and the metabolic interactions, predictions and so on. We will focus on metabolic interactions and exchanges in these communities because they are very interesting and they have a key role in the survival of these communities, the survival of organisms that drive these communities, and also the health of the gut and so on, because there are certain metabolites that these organisms produce like short chain fatty acids, which are very central to the health of the gut. You can read a little bit more about them in this book that you see on the right. It's a short monograph where we discuss in great detail a number of computational approaches to model microbial communities at a systems level.